Let me open the chat just in case that you write something. Okay. And now let me go to full screen mode. Can you see my screen?
Yes.
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 Perfect. So let me finish this. So once you receive the reads from the sequencer, you receive, depending of the methodology, you receive FASTQ files or maybe BAM files. No, PacBio is producing BAM files. It's a method of giving you, even if they are not have been mapped into anything, they like to store sequence as BAM. But yeah, the most common thing is FASTQ files. So once you have the FASTQ file that you will have here, it's important to rule at read processing and filtering. And once you have the read filter, of course, you can map the reads. You have an RNAseq analysis, or you can assemble them, or you can do other analysis depending of your goals. So this is a common step that is being used for a lot of different processes. So it's why I wanted to put this in this section. So I'm not going to go to here, but remember that I explained to you what is a FASTQ file. And remember that for the FASTQ file, you will have a quality score. And the quality score defines how good or bad was the sequence, the nucleotide call, okay, when this machine was producing the sequencer. So we want to remove those. So I add this extra one compared with the last presentation that it tells you, depending on the Phred score, the probability of the incorrect base and the base accuracy. So mostly when we are working with Illumina, we are targeting 30. And we are have this the probability of incorrect base of 1 in 1,000. Then the base call accuracy will be 99.9%. Okay, so we want to remove those. So that's will be done example here with a figure with a chromatogram. Of course, in next generation sequencing, we don't use chromatograms, but it will be an example. So, for example, this A that you can see here, this A overlap with this blue peak, this green peak. So that will be an example of something that will have a low quality. If you are thinking, for example, in Illumina, would be something like when you have some specific cycle, some specific plate, you will see kind of a reduction, maybe in the intensity, and it's not sure about the signal. So that is how these low Phred score are interpreted. So when you get these things from your system, you need to do several things. When you receive the sequence for the first time, you need to do several things. So first is ask you yourself if the sequencing produced the expected number of reads. So you was contracting something like 20 millions per sample. You need to check that you really receive 20 millions per sample. Okay. Then do the reads have the expected average length? With Illumina, for sure that should be a fixed length. But with long reads, you know that you have this variation. So the average should fit in what was promising. If you are doing your sequencing yourself, if you have an Oxford nanopore, of course, you need to check that, okay? And of course, if the reads has the expected nucleotide score, so for example, with Illumina, everything should be on 30 or more, no? So all these things, of course, are technology dependent. So once you have that, now what is the tool that you can use to verify that? So one of them is FASTQ stats, and you will use you with your exercise. So you run FASTQ stats, then your file. And you use the symbol greater than to redirect this output into a file. You will run this program on this file, and then the output will be writing in this file. So you will have a file that will contain some stats like read, number of reads, length, the maximum length, and so on. So that is an easy way to check that you have the quality that you are expecting or not. Other tool that you can use, and you also will use that during your exercises, will be FastQC. So FastQC have a graphical interface, but you are going to run this without the graphical interface, okay? And the thing that you will have is once you have the graphical interface or not, you have a file that will be a zip file with a directory with several files. So you open this directory and you have an HTML file. So you open the HTML file, you will see everything in your browser, no? So you will see different sections and then you can click in each of them. And you will have basic statistics as you can see here, but also you have per base sequence quality, no? So this is an example of per base sequence quality. So this is box plots, and this is the nucleotide of the different position from Illumina. So in this case, we have something like a length of 90. And you can see that at the beginning, everything is in the green zone, so I mean that everything is fine. All the reads are up to 38 or 30 or 32, so that means fine. But as soon as we go to the end, the quality of the sequencing is going down and down. And then you have some reads that have a quality below 20. When you clean the reads, so you compare one of these images before and after cleaning, you will see that after the cleaning, you should not see these sequences here, okay? Of course, depend on the technology. So there are the tools like these nanoplots that can be used also to evaluate the sequence. I'm not going to talk about that because in your case, you are going to use only Illumina. So that is evaluation. So what about cleaning and processing? So there are several things that you can do. So for example, cleaning and removing of low quality reads is something that is commonly done with short reads. Adapter removal is do mostly with short reads, sometimes with long reads, but depending also on the sequencing facility. Filtering reads below some certain length is something that is commonly applied to both short and long reads. Remove PCR duplications is also made done with short reads in assemblies. Remember that during the library preparation, you may have some steps of PCRs, so something that is done only for short reads and only for assemblies, not for transcriptomes. Removal of the contaminations can be done with short and long reads, and also sequence correction, also with short and long reads. So let's go to each of the different steps. So as I said, most filtering is made with minimum length and minimum score. So here you can see some guidelines that I usually like to give to people. So forget about 454, but with Illumina, usually you are going to use a minimum length of 50 and a minimum score of 30. Then with solid 20 and 30, ion torrent 50 and 20, and with PubBio and also nanopore, you only use minimum length, but no minimum Q score because you already know that these are quite low. Okay, so that is the common. What are the tools that you can use to do that? Some tools also remove adapters. So the most popular ones are the FastX toolkit, that is kind of all. utils, that is the ones that I like to use, and you will use it too. Prinsec is another one. It's a Perl script that is slow like hell, but has some applications that other tools may not have it. And then Climomatic is probably the most popular one. I especially don't like it, but it's not a bad one either. So the thing that you will use is you will use EI-utils in a fashion like this. This is for single reads, and you will be using that for parents, but you will see in the exercise some indications about that. So you will run this like FastQ-MCF. This is the command. Then remember, this is the first flag. Okay, so Q30, we are removing everything that is below 30. We are 30 in terms of Q score, sorry. L50, we are removing all the reads below 50 after that we clean the adapters. -O, this is the output that I'm going to give. I will give you some suggestion for the exercise too. The Illumina adapter file that contains the FASTA sequence that the program is going to use to search for the adapters. And finally, the input file. Okay, so that is how it works. These kind of commands. Contaminations can be removed in two ways. One of them is you can remove mapping the reads against a reference, that is your contamination. But sometimes if you don't know what are the contaminations, it's difficult to do it. Okay, so the common ones are E. coli and human, but of course you may have others that you even don't know. No? So you use bowtie or BWA to map these short reads, or BLAST-R and Minimap2 if you have long reads. And then it's special also to remove, well, you have an assembly, and you may have an assembly if you don't have a reference. You may do that. Well, yeah, I see one raised hand.
Sorry, I misclicked. My fault.
Oh, okay.
No, actually, I have a question.
Sure, yeah, please go ahead.
Okay, I have a question about the contaminations. Okay, you say that of course we have to clean all our data sets from contaminations, and you guess which contamination could be more common. Is that correct? Okay, but since I know what I am actually, what I want to sequence, why instead of cleaning everything that might contaminate my my sample, I just keep only the species I want?
And that is a good point. So it's the point that I wanted to make. If you already have,
okay.
So the thing. So you are doing, and you will do this with Arabidopsis, not with this experiment that you do. So if you know that your reference is Arabidopsis, then you are going to map your reads with your reference. And if you are mapping 70%, okay, you know that that's 70% that is map is Arabidopsis. And you forget about the 30% that doesn't map. So contaminations is something that you don't need to clean. It's the thing that doesn't map. When you know your reference, when you have a good reference, you don't need to be worried about contaminations because you are going to map against them. Supposedly that your contamination is not a plant, because if in your plant sample you have Arabidopsis and Brassica, that will be a problem. But also keeping in mind that you may not have the same mapping rate, still you can forget about that. But contamination is important. For example, when I was mentioning that you don't have a reference, if you need to do a genome assembly, well, a transcriptome assembly of this reference, of course you will need to clean the contaminations, no? And then is the second part, no? You can clean the contamination before you do anything or after that you do something. I always tell the same story, and it's starting to be an old story, but yeah, I always tell the same story, so I will do it again about contaminations. When I started doing bioinformatics, we were analyzing some tomato samples of a researcher from Florida. So we received the samples, and when we mapped the samples against tomato, because we were working with tomato, only 20% of the reads mapped. And that, of course, is a disaster. No, you always were going to have contaminations. A range that you can survive is, let's say, 30% of contamination is top. No, if you have more than 30, it's because you have a big problem. So, of course, we could map only 20%. So we have an 80% of contaminations. So was okay. So what's about the contamination? No, so we have a high percentage of contamination, so we go back to this guy and say, well, you know, we have a good news and a bad news about the contamination. Okay, so tell me about the bad news. Well, the bad news is you have 80% of contamination on your sample. So that is bad. That is bad. So okay, so what is the good news? So well, the good news is that the 80% of the contamination is a human contamination, so we can identify the person who did the contamination and even run some genetic analysis for the same price, if you want. So yeah, so of course, that was the story about the contamination. No, someone sequenced, so that we sequenced the technician that was playing with the plants. So was the story. Now, of course, we didn't publish the paper, but we were able to play with the data a little bit in class, no? Saying, okay, so this is the technician DNA, and this is how it look like. So, okay, so that is about contaminations. Recorrection is something that you are.
Thank you.
Oh, yeah. So recorrection is something that you are not going to perform from transcriptomes, of course, but sometimes it's useful too. So as I told you before, you may have reads that may have some errors. So a good way, I can see someone in the chat. One moment. No. Yeah. Okay. So you may have some nucleotides that are errors. So a good way to fix these errors is to just compare the reads between them. And then once you compare the reads between them, like you have in this case, fix it. No? So you have these reads that are the same. You can decompose them in k-mes. I'm not going to explain that specifically, but you can compare them. And then, for example, you vote them. So if you have 10 of these, one of these, one of these, one of these, you know that the 10 is the majority. 10 and the majority. So then you can go here and, for example, fix it. No? So this is the consensus. So if you have, for example, here a C, you should change this C to a T. Again, not important. Did you have problems with the some kind of background noise? If you have hearing a background noise, I guess that at some point it will be me because my computer is overheating, but I hope that it will survive until the end of the class. Okay.
No, Ari, it's just if two people have the microphone on at the same time, there's either weird feedback or your sound comes muted. So if someone asks a question, they should unmute while they hear the answer.
Okay. Okay.
Oh, I'm sorry. I am having poor audio quality, but I'm on Linux. Maybe it's for that reason. I don't know.
Yeah, please. Silence. All of you silence your microphones if you are not asking questions.
Okay.
So, yeah, recorrection, you have several tools. I'm not going to talk about it, but if one day you need it, you have here a reference. Recorrection can be done in two ways. You can compare the reads with themselves, or you can map reads to this sequence and then correct them. And again, I'm not going to go into much because it's a process that normally is not used for transcriptomes. Okay, and I think that that is the last. That is my last slide. Okay, so that is in terms of read processing. Questions that you may have about this part?
Actually, I have a question about the sample preparation after the RNA extraction.
Okay.
Of course, you have to reverse transcriptation, and you can use either the random primers or the oligo-DT.
Yes.
But is there any differences? I mean, the random primer, they should cover everything.
Yes. The problem with the random primers is it's very simple. When you are extracting total RNA, you have 90% of your sample is going to be ribosomal RNA. And of course, if you are working with an eukaryotes, you know that your mRNA is going to have a poly-A. So you have two options. You are not interested in the rRNA. rRNA is a contamination for your expression studies. Okay, so you are not interested in your rRNA. So you have two options. One of them is you purify the mRNA before. So for example, one way to purify the mRNA is with oligo-DTs and then depleting, get the mRNA. Other option that is the most common one is you use oligo-DT and it's going to be only transcribe the mRNA. So, but if you use random primers and you have a rRNA contamination, the problem is that this rRNA contamination is going to be transcribed too. And then when you are looking into the expression, maybe 20%, 30, 40% of your sample is going to be rRNA. And of course, because you need to normalize with all your rates, it's going to mess up your expression. So it's the reason why random primers could be used, and it's still being used in some occasions, but you need to be careful when you use them.
Okay, I see. But in my case, since I study the chloroplast, and the messenger lacks the messengers lack the poly-A. So in that case, I am forced to use both, or just one.
Exactly, exactly. So you have exceptions, no? In the case that you are studying the chloroplast, you have to use random primers. So in the case of in your case, the thing that you need to do is to use a depletion rRNA method if you are not interested in the rRNA. So depletion methods, for example, are sometimes probes that can bind to the rRNA and get the RNA from. And you have depletion methods for most of the model organisms. If you are working with something that is not a model organism, then you need to assume that it's going to be difficult to get to separate the mRNA, because in this case, as I said, your mRNA is not going to have polymerase.
Okay, thank you very much.
Okay.
Actually, can I just ask a quick question?
Yeah.
Where do you find and how do you select references? Since we talked about this a bit, but I don't really know where I can find them, basically.
Oh, you go to a database and then you check if your genome of interest is there. So let's suppose that you are working with human genome. In human genome, the reference is going to be NCBI. So you can go to NCBI and download the last version of the human genome, that is the 3.8, I think. So that is NCBI. Mouse is the same thing. Mouse, the place is NCBI. If you are working with an unknown species, the problem is that not everything is in the same place. So NCBI may or may not have your reference, or then you need to go to a specialized database. So the thing that I'm proposing to you, if you don't have any idea, I mean, the three places that I will recommend by default will be NCBI genome is one of them. Phytosome, if you are working with plants, is a good one, or ensemble genomes. If you don't find, still you don't find your reference there, you may just Google and see if in Google you will find your reference. And of course, the last thing that may happen is that sometimes you see that someone has published a paper with the genome, but the genome is not in any place. And that happened too, sometimes. So in that case, you can send an email to the authors and say, well, you know, you have published a paper, your genome should be available. Can I have access? If they are nice, they will send you access to the genome, or sometimes not. No, I have all the different possibilities.
Okay, thanks a bunch.
Yeah. Okay, so I'm going to keep this section as closed. So now, okay, start recording.